Okay, so the cell wall is the border between self and, no, we don't have the loudspeakers.
The cell membrane controls traffic into and out of the cell and surroundings.
Doesn't work.
Like all biological membranes.
This doesn't work. Sorry for that. No movie. So, prokaryotic cell, essentially one or two membranes around it, and everything else is not membrane. While in the eukaryotic cell, you have the outer membrane, you have a membrane, bless you, you have a membrane around the nucleus, you have various membrane-bound compartments, such as the mitochondria with two membranes, and many vesicles, many other things. Ultimately, the cell wall is the way in which you separate out self and the world. The cell wall is essentially somehow the analogy of a medieval castle. This is the medieval castle, that's the outer wall that defends the city from the outside. What is it built of? Lipids. Why do you use lipids? Okay, in a medieval castle they use stones. Why don't we use stones?
Too sturdy.
Too sturdy, yes. Not flexible enough, not organic enough, not chemical enough. Stones are too physical. Right, lipids. So what are the features of lipids? Again, what else is great in lipids that may be interesting here?
You can aggregate them easily.
Yes, you can aggregate them. Okay, that's a great point. But let me just, so you can layer and stack them, you can form them very well. So let me just give you an alternative concept to that. Why not build a salt crystal around it? Would be so one thing is defense against attack from the outside. A layer of salt around the cell would be great to defend against intrusion.
It seems quite unflexible.
Salt is also soluble. Won't go well.
That's an important aspect, right? What you have inside, in some sense, or outside, you have a lot of water swimming inside the cell and outside the cell. So we want to have something, and lipid, in fact, is perfectly wonderful as a protection against water intrusion. Salt wouldn't be that way. It would lack somehow the flexibility, possibly. Maybe we could still have flexibility with salt in some sense, but here's another thing, that is the soluble. And in fact, again, you said that already, so there are two layers of lipids sticking in. The head groups are on the outside, the non-head groups are on the inside. When I look at all these cell walls, they're made out of lipids. Are they all the same lipids? All the cell walls? No. So what do you know about the lipid composition of different cell walls, about prokaryotes, eukaryotes, about different eukaryotic compartments? You know very little. This is fair enough. The world knows very little. It's one of those detailed research topics. There are remarkable differences. And in fact, the differences are even such that within a particular cell wall you have differences. You use different lipids in terms of chemical nature of the lipids. Now in this particular case I would really call it chemistry for that. So those are lipids that have different chemical features. But this is not well studied. Mostly the assumption is always on the very, very coarse-grained level. Lipids prevent water from going through easily. Lipids are lipid and the whole thing is lipid, full stop. At two layers, but ultimately this is the way to put it. The protein inside is hydrophobic typically, outside is hydrophilic, and in a membrane protein that is inverted, right? Now another issue of the membrane is it is actually very simple if you put something through, like a wire or a helix or something, to move it in that direction is relatively simple. Okay? How is it to move it in that direction? That actually is very tough. Why would that be the case? This part. The first part I can actually not answer. Why lipids are so easily movable in that case. I believe it's not a problem. I believe it's ultimately the answer. It's a feature. I don't know the explanation. But this is clear, right? Why? Now, is it obvious?
No, because if you want to tear it apart, basically you have to introduce water to the hydrophobic part, which doesn't really work because biophysical.
Well, but what about you? This is a hammer. It's not very. I don't put an ice hammer there. So you could imagine to hammer it with something that is actually not water.
Yeah, but then like you hammer within the water, right? The water will kind of get pushed through it and just.
No, this hammer is not water.
No, like if you hammer it, like there's some opening to the water, and it wouldn't like energy could very
easily flow. So we have good reason not to want that. Yes, that's true. So you don't want to have intrusion. So that's why it's a good feature that this is not the case, and this is good enough. So is there anything that you believe you could, like a hammer, boom? Yeah, you know, a real hammer. No, but on, something on the molecular level. Are there things that just hammer inside the membrane and then...
hydrophobic stuff.
Yes.
Hydrophobic? Stuff.
Yeah.
So small molecules. But they're not really hammers, right? They are sort of swimming, sneaking up on the membrane and slowly going in there. Ideally actually finding a receptor. And that is essentially the heart of the transmembrane prediction. So typically the way to cross the membrane is through membrane proteins. That's why we need them. But can you imagine something that is sort of a little more radical than membrane proteins?
Detrogen.
What?
Detrogen.
Well, how would that do it?
You could basically...
It's chemistry.
It's chemistry.
Yeah, it's the same thing. It's a toxin. And it's like a toxin. So you sneak up on the membrane and you slowly sort of get in. Typically, again, through membrane proteins. But no, I'm talking about something else. Yeah?
What about viruses?
Yes. How do viruses do that?
Actually, I didn't know.
Actually, they find membrane proteins. They are these sneaky bastards who find the same mechanism of transport that anything... by the way, why do we need the membrane to be somehow semi-permeable? But before that, do you have a question, or...?
No, it was the thinking about
Yeah?
the definition of hammering.
So I'll get back to the definition of hammering in a moment. But before we get into the definition of hammering, this is a very good point. You cannot answer that question before you address that question. But before we get back to the hammering, why does it have to be semi-permeable? Why do we need to get things through? Why is the crystal not such a great idea? Or well, okay, there we have different reasons. Why wouldn't I build an iron wall around it, for instance? Iron is actually cool for the medieval villages, but
But the villagers inside somehow have to live. So
they need stuff inside and they put stuff outside. So there must be a constant exchange of material. Energy typically in, some other stuff out. So we need something that goes through in both ways, and that is exactly what viruses misuse. Now back to the definition of hammer. What I mean by hammer is somehow the idea of mechanical energy that actually like cracks open a hole into the membrane, and it does it.
And for it to stay open.
Then ultimately it will close again. And one actually sort of positive feature of the fact that you can freely move in that direction is there is a free ability to close holes again. In some sense, this is one of the aspects, but you most likely could do that differently. But I mean some rapid energy. And here's an answer. Unfortunately, I don't have a movie about that. Marek Basler just showed me this 2 days ago. I was totally fascinated by it. So it's the bacterial injection needles, the so-called type 6 secretion system. The type 6 secretion system, so this is all built up of proteins, and then on top you have this long extension of molecules that in fact get very long, and the way these are bound, there are some salt bridges here, but the way they are connected is not really through the salt bridge. So the salt bridge could form, but in the shape that you see here is not formed. Then you begin to form a salt bridge. Upon the formation of salt bridge, you release energy that makes the other salt bridges form, and the whole thing collapses. In this collapse, you actually have material in there that is shot through this. It's a needle. You don't believe it, huh? He's amazing, I tell you. This is totally amazing, and I'm afraid that what we have here is a good example why the membrane has to be protective against hammer attack. Okay, membrane helices. So this is a typical membrane protein here. These are others. Can anybody see the membrane? So again, there is no membrane that you can see in the sense that you, but do you imagine that you see the membrane? By the way, you do not see the membrane in these crystal structures because lipids are not in the crystals. Where exactly the lipid bilayer is, we do not know. But can you sort of see them? You can? So you sort of see that the helices align here, right? And there's in all of these places, you see something like that here again. And you also see that there are different arrangements. So we have sensors, holes, we have things that are completely closed and hooked up. We have in fact what the membrane anchor is called. So this is transmembrane, these are transmembrane. Here you would have a helix that would be sort of anchored. It hangs on the lipid. Again, if you sort of imagine that you see the lipid, then the lipid layer is somewhere here. And the blue helix here clearly is not going through the membrane. Is also not like the red one here that is going completely into the protein. This one seems to be aligned with a layer of the membrane. So in some sense it is anchored to the lipid, and that is also something that we observe. Now these are some different types of membrane proteins. There are many out there, but ultimately again you see there are always helices that cross the membrane, or helical membrane proteins. When we look at comparative modeling for membrane proteins, I told you, well, so half we can do by comparative modeling, and this is a little bit too pessimistic view. And I told you that the power of comparative modeling is increasing by not doing anything, simply by waiting for the databases to increase. Now when we do comparative modeling for membrane proteins, how do you believe this will look? So say we can do comparative modeling today for half of all the proteins. For what fraction of membrane proteins do you believe we can do that? It is less, indeed. That's the fraction. It's much less. It's this big blue thing here. Unfortunately, for that, just sitting there and waiting is not really making a big impact on the story. So today, this is a while ago, so this is because there are very, very, very few membrane proteins in the PDB. We believe that in the ballpark of 20% of all proteins are membrane proteins. We will get back to that number. But why is it true that if there is only 20% of all the proteins, if about 20% of all the proteins are membrane proteins, why are they only 2% of all the structures?
It's hard.
It's very difficult to do. Do you have any idea why it's difficult to do?
They're stabilized by the membrane, and when you crystallize them, you have to remove it, so they just lose their population.
That's really, really mean. So you have something in a native environment, you take it out of the native environment and you completely deprive it of the stabilization. So people are trying to sort of create artificial membranes, to in fact put them through artificial membranes and put them into that crystal. That's one way of doing it. Then there are other ways of doing it. There are some membrane proteins that exist in so high concentrations that essentially you scoop them out of the membrane and then you already have a crystal. This is slightly overstated, but somehow close to truth. One of those is called the photoreaction center. Does that ring a bell with anybody? Does anybody know photoreaction center ever heard? So photoreaction center was in fact the first membrane structure that was determined, and in fact by a group in Munich. Now, in terms of prediction, or there's another word that I want to introduce here, the word topology. I introduce it because it's used and it's totally misused. Topology may raise all kinds of ideas. For membrane proteins, all that this word means is the orientation. So the topology is something that has two states in the membrane field. There's out and there's in. And whether you're in or out depends on where you're at the beginning of the protein for your N-terminaluses. Is it outside of the membrane or inside? Okay? That's all. But this is a very important classification. Now, functionally important classification. When we look at drug targets, then about 60% of those are in the membrane. So that's a high fraction. And many of these are so-called GPCR receptors. Sorry? Okay, here is an example. 40% of all the drug targets, in fact, are GPCR receptors. And they have 1, 2, 3, 4, I believe I got the orientation wrong, 5, 6, 7 helices that sit like that in the membrane. And they have a particular orientation. Here's another type of membrane proteins. Those are, this particular one is the voltage-gated potassium channel, where you see that these two parts of a chain come together and they actually regulate how the potassium ions, one ion at a time, is passed through that protein. And they are specific to potassium. How do they achieve that? By the,
Size, maybe?
By exactly the size. It's the size combined with the local physical charge. So we're talking about moving charges. So this is in fact something, when you look at it, it has an attractive potential. And then you regulate by size. And in fact, on top, it's an active transport. So they don't just fall through. They essentially, there are photos of that where you see that essentially it's like the arm moves the molecule to the next, to the next, to the next. And they lie there and waiting. Remarkable structures. This is the one from McKinnon that got the Nobel Prize. That's a remarkable structure. Okay, in terms of transmembrane prediction, it's a membrane helix. Okay, so why not just use, we talked about secondary structure prediction methods, it's a helix. So we can do secondary structure prediction methods, right? And that's what we get. We apply a simple secondary structure prediction method to the membrane helix here, and we get something that is not quite right. It's a little bit more wrong than right. Now, the reality is that if you had that sequence and you put it into a globular protein, maybe it's going to be a strand if it has a next strand next to it. But since it sits in the membrane, actually it turns out to be helix. And the method that predicted this strand here never knew about membrane environments. So somehow we have to retrain. To account for the fact that in a globular protein, hydrophobic is in, hydrophilic is out, and now we have to reverse that. So somehow we have to look at the other way around. So the other way around means hydrophobic residues are in the lipid. So the simplest way of doing that is identifying hydrophobic residues. there is, for instance, a hydrophobicity index by David Eisenberg. One of the early ones is from David Eisenberg, that simply looks at the amino acid and measures in some environment how this translates to hydrophobicity. When I look at two different hydrophobicity indices, on this particular slide I actually have 1, 2, 3, not 2. It's Kite Doolittle, Eisenberg, and GES scale. The 20 amino acids. This is the index, and you clearly can see that not all lines are absolutely identical, or not all bars are absolutely identical. So in most cases, you have the type of bars typically go in a similar direction, but then we have already here a big difference. So between this one and that one is a large universe in between. It's essentially two opposite sides of the spectrum. Now, when we look at more, and now we sort of are here in this graph, I normalized it for visibility. I understand this is very difficult to make my point that this is easier visible than the other one. But it's also true that we have now 5 indices here. And again, there are some similarities, there are many differences, there is clearly no one signal. Now what we could do is we could cluster, right? We could take all the hydrophobicity indices and cluster them. That's work that has been done by a Japanese group, and now this Tomi Kanisha in 96. And in 96 they actually clustered 402 indices. 402 different types of indices. They began with a lot of hydrophobicity indices, but then they pulled in to get to this large number also other things. And again, so the good news is hydrophobicity indices cluster. So in the universe of all the indices, this is hydrophobicity, and this is the largest cluster. But the bad news is some things such as beta propensities that are actually something totally different. Alpha and turn propensities somehow have overlap, and there is still an immense spread. So the idea that membrane is hydrophobic, so find the hydrophobic residues, is very simple. But the solution is not that simple, because indices differ. What else could we do in order to predict membrane protein structure? So we could look up hydrophobicity, and I'll get back to that in a minute. Just pick one index. Okay, let me begin with pick one index and ask my question later. So pick one index would be something like this. By one particular index, you would call all of these here hydrophobic residues, and then you would possibly say this is my membrane helix. And as you see, this would be a very correct prediction. Now in this particular case, I have isolated non-membrane helix. Maybe I join them and make one long helix. Again, for this particular example, that would be the right thing to do. And this sort of idea in the next level came from Gunnar van Heijnen, who is one of the giants in the field, and in fact is also, here you see him announcing the Nobel Prize in Chemistry in 2008, is the Secretary of the Nobel Prize Committee for Chemistry, the current one. Now, he was the one who pushed the prediction of membrane proteins more than anybody else, and of single peptides. He contributed immensely in the early days, still contributes immensely to understanding of membrane proteins. And originally, really here, this is only hydrophobicity. Now, in the next, so one issue for hydrophobicity is that somehow you have to make a cutoff. Say, if it's higher than that, I call it hydrophobic. Call this region here, for instance, the membrane helix. Lower, below, I clearly call it not. And then in between, and that already is sort of a great idea, in between I say, maybe this is where I will elongate my membrane helix to make them fit more to what I observe. And so this is the hard threshold that will give you these membrane helices. From these membrane helices, it immediately would sort of leave some out, because this is way too short for a membrane helix. It just cannot span a membrane. It has a certain length. The certain length, in order to do a typical membrane helix, you have to have something like 15 to 25 residues. So this one here clearly cannot happen, right? This one cannot happen, this one. So you can remove some. In the next step, you could argue, okay, this is now my solution of those two. But now comes the sort of second level. And the second level, we may find some reasons to make it longer. And we may even find some reasons to, in the second level, in fact, long enough would be those 5. Okay, we have the extreme model where we have only 2 membrane helices in the higher level, and the lower one where we have 5. How can we distinguish which one of those two is better? Well, we could fit a distribution of helices, right? What else?
Normally that some for like the tail is outside or inside, so there can't be a helices at the end of the sequence.
So this being biology, most of the time the beginning of the protein is not in a helix. In fact, for many cases the N-terminus is used as a signal for secretion, is used for building a signal peptide. That is very hydrophobic, and now we get into the crux of the story. Part of that is actually chopped away and sometimes inserted as a membrane helix. That begins very much with a mature, so the final, after the cut, it is in fact the N-terminus that then is inserted. So this doesn't quite always work, but in many cases it does. The C-terminal, there are in fact also hydrophobic signals on there, but much fewer. So mostly the C-terminus is not in a membrane. But again, there's a probability signal, just in the same way in which in a standard protein N and C-terminus typically are not helices and strands. In many cases they are. Similar distribution. Okay, so how could we distinguish between these two different models? And so I'm going to introduce some clever averaging that would sort of change the probability, and ultimately got back to the idea of the topology. And the idea of the topology is, so I introduce another concept before I address the question that I phrased before, and that is the orientation I said is the topology. And what he found out is these two distributions. That's the periplasmic loop, and that's the cytosolic loop. So this is the inside, that's the outside loop of a membrane. So inside, outside. Yes, the colors are right. Number of positively charged residues. The number of positively charged residues is simply K and R. So it simply counts R and K. And you see that there's a prevalence in here, and he coined that the positive inside rule. So positively charged residues are more often found at the inside. Now when he says inside, he's not meaning the entire globular inside domain or region or protein. What he means is really just next to the membrane helix. There's a little bit of a loop, a little bit of an extended region, and extended is also the wrong word, a little bit of a region that is not immediately another globular domain. And when you look at those residues, they are enriched in positively charged residues with respect to the outside, with respect to the loopy regions on the outside. Okay, this is clear in this graph. Now, since that is the case here, you can now in fact use that to distinguish between these two alternative scenarios here and pick the one that makes the signal of the positive inside rule better. So if 5 helices gives you a better positive inside rule, you predict 5. If 2, then 2, and something in between. You simply optimize the choice of the threshold in between. You optimize by the charge. The idea works. The method is no longer used that well because there are better methods out there. But the idea is a very simple and important idea. That's why I present it. So it's also, I rephrase the same idea here. So you simply look at the predicted membrane helices, you count, for whatever your model is, you count the number of charges, look at the difference here, and then in this particular case you say first one is out. Okay, let's back to this one. Now, okay, gonna essentially use some hydrophobic, some criterion, some scale for hydrophobicity. What else could you do? So somebody does an experimental scale for what is hydrophobicity. You know that hydrophobic residues are on the membrane. You use that scale to predict membrane helices. On a similar level, without any big bells and whistles, as simple as that, but in a slightly different way of thinking that is more prediction oriented. How could you do that? How could you accomplish something like that?
Structures of known helices and try that this model just predicts future liquid sequences.
Yes, that is what you can do. That's essentially exactly what you do for secondary structure prediction. So you now have a secondary structure prediction that is specific to membrane helices. Now, what I'm saying here is the idea is to optimize hydrophobicity scale for prediction. So you derive a hydrophobicity scale that is optimal to help you in prediction. Now my question to you is, what you said is you derive a probability for a particular residue to be in a membrane helix versus not. And here I say we, in fact, we decide upon what we call hydrophobicity scale to optimize prediction. Are these two different things? I have to, does anybody remember, some of the people were in the room when 17 years ago, how many days ago, I can't remember. At some point I introduced this idea of a website where you can vote on it. So you all had to log, does anybody in the room remember? Does anybody remember what that website was? I do remember that some people didn't like it, but I still want to try it again. Maybe there's an incentive for the ones who didn't like it to not remember. I have a very high incentive and still don't remember. So the advantage was that we got a higher participation in these yes-no votes when nobody knew who said yes and no. So, is that the same as a probability for predicting membrane helices? Yes or no?
No.
Yes. Yes. Yes.
Yes?
No.
Yes?
Thank you. I was about to go to say this is men. Oh, yeah.
I say yes.
Yes.
Yeah. Majority is always right? Absolutely the same. You just put different words, but ultimately, and again, this is one of those examples that was not clear in the publication. It's not quoted, that's not good. Oh, it's quoted up there. And it had not been clear to the reviewers either. And in fact, there was another idea underneath here. And the other idea is that you could essentially call something that's a probability for prediction hydrophobicity scale. So that's sort of an inverted way of looking at it. But in principle, it's exactly the same thing, right? So when you do something like that, the next question is, what else could we do? Well, what I'm fishing for here is to simply use alignment information, right? In this particular case, I show the alignment information with a neural network, but you could use it in other ways too. So where in fact, this is the story from globular secondary structure prediction. Now the output would be different. There would be membrane, not membrane. We still would use at every single position the sequence alignment information and essentially get this solution. In the secondary structure prediction method that I presented, we had this issue that membrane helices were predicted to short, because in training neural networks you never saw the correlation between I and I+1 or I+5. And that was solved by having sort of two levels of neural networks, where the one predicts from sequence the membrane place, and the next one sort of takes the prediction as input and sees that there are correlations between adjacent predictions. Now, and this in this particular case worked very well. Now, how do I predict whether a protein is a membrane protein or not? What this one does so far, it predicts that a particular residue is a membrane residue. Next question. How do I distinguish between proteins without membrane helices? How can I use that to break proteins with and without membrane helices? So far we have created a method that predicts for every single residue whether that's a membrane helix or not. But now I want to estimate from my prediction how many membrane helical proteins do you find in human? How do you do that?
Just by looking if the helix could be found.
So I find no residue in a helix and I call it not a membrane protein. That's true. That's an extreme way of doing it. But can you imagine what would be a problem of that? So let me show you a typical, I misunderstood what my next slide is. But let me, although I give this the wrong order, or the non-intended order, I can still make my point on this one. Say this were my profile for transmembrane. Okay? Now my next statement is I would have to somehow put my helices in there. I still, just like before, have to find a threshold. So the threshold could be this, or 0.5. Or the threshold could be whenever this, for transmembrane and not, whenever the T is higher than the N. So there are different ways of playing this. But you see that, well, if this is higher than 0.5 than T is, so they are the inverse images of each other, then T is higher than N, the way they are drawn. And you immediately see that this is actually too long for a membrane helix. No, you don't see that. Because I didn't give you any. This is 25, 50, 7500, I guess. Oh, this is 50. No, this is 50. So they are way too long. So now you have to somehow still find the right helix length in there. But if my scale would have been slightly lower, ultimately my point is the best way to distinguish whether a protein is a membrane helix or not is you simply find the longest thing that looks like a membrane helix and look at how strong that is. Okay? So we will get back to that. My next question is, how can I use now this profile to find the membrane helices? Profile is the wrong word. It's a simple, and actually I'm going to stop here. This is for next week. Thank you for your attention.